Today we'll be talking about microbes in the world around us. So what are microbes? Microbes are single-cell organisms so tiny that millions can fit into the eye of a needle. Understanding these is vital to understanding the past and the future of ourselves and our planet. Microbes are in the air we breathe, the ground we walk on, and the food we eat. They're even inside of us. In general, microbes are great. If it weren't for microbes, we wouldn't be able to function as we do, nor would our planet be as it is today. On the other hand, they are bad in some cases in which they take forms in infectious diseases. Hygiene hypothesis really has nothing to do with your personal hygiene. It describes your microbial community and how abundant and diverse it is. The more diverse and abundant, the less likely you are to have allergies later on in life and be prone to problematic symptoms with health. Now, using the hygiene hypothesis, we form our own hypotheses in order to further investigate this subject. Our first hypothesis states, being born naturally will increase the number of microbe species. So we ask, were you born naturally or by C-section? Our second hypothesis states that growing up in a rural area will increase the number of microbe species. So we ask, did you grow up in a rural or an urban area? And for our third hypothesis, it states, more time spent outside per week will increase the number of microbe species. So we ask one, how much time do you spend outside per week? With our hypotheses in mind, we move on to the methods section, in which we demonstrate how we obtained our samples and applied them to be further analyzed to gain results. The first requirement was for each student to take a survey. These surveys consisted of questions asking the student about their past and present environments. Next, to obtain our samples, students need to swab a body part. Half the students are to swab the inside of their belly button, while the other half the inside of their forearm. These swabs are then taken and put into a solution, removed, and then that solution is put into a 32 well plate to be further analyzed. Each of the 32 wells is associated with a different carbon source commonly found to our microbes. We allow 7 days of incubation in order for the microbes to interact or not with the carbon sources before further analysis is needed. After the incubation period is over, we put our well plate through a complex machine that uses light absorbencies to determine whether or not the carbon sources were used up by our microbes. This data will also be put through an ANOVA analysis to determine whether or not we have significant results. For our results, we compare species richness to the three previously stated hypotheses earlier. Species richness is the carbon sources that were actually used up by our microbes against the carbon sources that were in the population sample. To make things simple, we are looking at a p-value. P-values show how likely something is to happen just by chance. Anything less than 0.05 or 5% is considered significant. In the instance of how we were delivered, 0.33 or 33% shows that this is non-significant. Here we are comparing species richness to the environments from which we grew up in. We can also see a p-value of 0.20, 20% likely due by chance, so we also say that this is non-significant. And for our final hypothesis of time spent outdoors per week versus species richness, we have a p-value of 0.07, 7% likely due by chance, so again, this is not significant. We wanted to investigate how the environment affects our microbial communities in a variety of ways. We posed hypotheses, obtained our samples, and analyzed results in order to see whether or not our posed hypotheses were significant. The results show that how one is born, the environment one grew up in, and the time spent outside per week does not have a significant effect on one's microbial diversity. The p-values were all greater than 0.05. Therefore, our hypotheses were all rejected. Other studies have shown that there is an increase in microbial diversity due to these factors, but ours did not. We could possibly attribute this because of a small sample size and variance among participants.